The overall goal of the following experiment is to develop a phenotypic screening assay that quantifies botulinum-mediated cleavage of its substrate SNAP25 with a view towards identifying small molecules that inhibit toxin function. During normal nerve signaling, the action potential causes SNAP25 to bind the synaptic vesicle to the plasma membrane of the motor neuron terminal, resulting in the release of neurotransmitter. However, when botulinum neurotoxin is present, it is taken up by endocytosis and released in the synaptic bouton, where it cleaves SNAP25 and so interferes with the release of neurotransmitter, causing paralysis. This assay identifies molecules that can inhibit botulinum toxin activity and thus prevent SNAP25 cleavage in the presence of botulinum toxin, and so restore neurotransmitter release in the neuromuscular junction. A high-content imaging assay performed with mouse motor neurons is used to demonstrate the preservation of SNAP25, resulting in the identification of small molecules that can inhibit the metalloprotease activity of botulinum neurotoxin A.
The main advantage of this technology over existing methods like cell-free biochemical assay is that multiple phenotypic endpoints can be captured simultaneously from a single well containing motor neurons treated with screening compounds and toxin.
This method can help answer key questions encountered during library screening, such as quantification of cell-based activity and cytotoxicity.
The implications of this technique extend towards therapy for botulinum intoxication because it can identify